Hi, back with another session here. Here we'll discuss about disorder of sexual development, that is DSD. So before going to the main topic, I'll first discuss here anatomy, something about development of male and female gonads. So SRY gene is very important for development. If like SRY gene is present, then male organs will be formed, that is testis. If SRY gene is absent, then female organ will be formed, that is ovary, and both will be formed from the genital ridge. So in the male, if like testis is present, then FSH will act on the Sertoli cells and LH will act on the Leydig cells. If on from the Sertoli cells, this Mullerian inhibiting factor will be released, or also known as anti-Mullerian hormone. So by the name it's clear that it will suppress the Mullerian duct. Then only the remnants of the Mullerian duct will be left in the male child. If on the Leydig cells, from the Leydig cells testosterone is released. Okay, so testosterone is actually important for this development of the Wolffian duct. So basically two type of ducts are there. One is Mullerian duct and the other one is a Wolffian duct. Mullerian duct will lead to formation of the female organs, and Wolffian duct will lead to formation of SEED, which is a male organs. We'll discuss it later. So the testosterone will help in the development of the Wolffian duct, that is male internal organs will be developed. DHT is dihydrotestosterone. So from testosterone, 5-alpha reductase enzyme will act and leads to formation of dihydrotestosterone and leading to development of male external organ. So why SRY gene is important here, we will see now. Like if SRY gene is absent, then female will be formed, female ovary will be formed, and from this ovary no Sertoli cells are there, either no Leydig cells, so no Mullerian inhibiting factor, then Mullerian duct will be developed as normal and female internal organs will be formed, including the uterus, fallopian tube and the upper part of vagina. No Leydig cells, that means no testosterone. No testosterone, so where the 5-alpha reductase will act. So no DHT. As no DHT, so female external organs will be formed, and also there is no suppression of the the suppression of the Wolffian duct will be there, and remnants of Wolffian duct can be seen in the females. Okay, so we'll now go to the topic disorder of sexual disorders. So first we will take overview. Disorder of sexual development involve a congenital discrepancy between the presence of male factor in XX and absence of male factor in XY. So default genital development is female. As you can see, if SRY gene is absent, then there is 100% chances of being a female. So default genital development is female, with male development requiring testosterone, DHT and Mullerian inhibiting factor, MIF, that you can say. And key diagnostic test to detect it is XX or XY is karyotype. So DSD involve chromosomal sex, that is difference in abnormalities in XX or XY, gonads, testes and ovaries, or maybe the external genitalia. So here again we will discuss that Mullerian degeneration, it is because of this Mullerian inhibiting factor. So starting from the here testis, if SRY gene is present, testis will be there, Sertoli cells, Mullerian inhibiting factor and Mullerian degeneration. If testis is present, Leydig cells, testosterone will be released, dihydrotestosterone, external genitalia, penis, scrotum, prostate will be formed, mesonephric ducts, internal genitalia, seminal vesicles, epididymis and vas differences will be formed. So this is all about the male development. So there are varieties of disorders covered in the DSD. First, there is type of DSD. It can be XX DSD and XY DSD. But if you will see from the 6 chromosomal disorders, there will be aneuploidy of 6 chromosomes. Aneuploidy means there is euploidy is a normal number of chromosomes. If XN is there, that means there is abnormality of the number of chromosomes. So it can be a Turner syndrome, that is 45X, Klinefelter syndrome 47XXY, or double Y males X double Y. So Turner syndrome will mostly affect the females. Okay, so it will affect the females, leading to a short stature, lack of ovarian development, and the other physical abnormalities. You all know streak ovaries. Klinefelter is males with an extra X chromosome, so leading to small testes, infertility, tall stature, and learning disability. In double Y males, X double Y, normal fertility, and male external internal genitalia, either of each, and associated with the tall stature, acne mostly during adolescence, and learning disabilities, behavioral problems can be there. So types of DSD can be XX DSD and XY DSD. In XX DSD, by the name it is clear in the females, so presence of ovaries with ambiguous genitalia. Exposure of XX individual, generally when excessive androgen will be there, so it can lead to CAH or gestational hyperandrogenism. Fetal vulnerability occurs between 7 to 12 weeks of gestation, because mostly the gonads development and the this sexual development is there in between the 7 to 12 weeks period. In XYDSD, presence of testes with ambiguous or female external genitalia, caused by underexposure to androgens due to conditions like gonadal dysgenesis, 5-alpha reductase deficiency, androgen insensitivity, or testosterone synthesis defect. So one by one we'll take all of this. First we'll take this XX DSD. So XX DSD is due to disorder of androgen axis. Okay, so XX DSD is due to disorder of androgen axis. First will is congenital adrenal hyperplasia. This is the most important topic among the all. So congenital adrenal hyperplasia is CAH. Most common causes 21 alpha hydroxylase deficiency, 90% of the cases. Okay, so we'll discuss this in a detail.